This week, I will be reviewing two papers on the sublethal effects of oil spills. In this review, we will be looking at the research by Carlos Alonso Alvarez, Ignacio Munilla, Marta Lopez Alonso, and Alberto Velando on the sublethal toxicity of contaminants from the Prestige oil spill on yellow-legged gulls. Larus Michaelis. This research, published in 2007, was conducted as a concerted effort by the Ecology Unit from the Research Institute for Hunting Resources in Ciudad Real, Spain, the Department of Ecology and Animal Behavior at the University of Vigo in Pontevedra, Spain, and the Department of Animal Pathology from the University of Santiago of Compostela in Lugo, Spain. This research was published in the journal Environment International in 2007 and has been cited by 51 other articles. In the paper, researchers wanted to study the effects that toxic polycyclic aromatic hydrocarbons released from oil spills have on the health of yellow-legged gulls. They tested for the presence of 16 polycyclic aromatic hydrocarbons in both adults and chicks, in oiled and unoiled colonies 17 months after the Prestige oil spill. They used this data to establish spatial differences in the concentration of total polycyclic aromatic hydrocarbon pollution. They additionally recorded plasma biochemical parameters, hematocrit, and body mass of individuals to correlate the concentration of TPH levels with overall health. Lastly, researchers compared 4 blood plasma compounds and body mass obtained before the Prestige oil spill with levels after the oil spill to observe the before and after impact of the oil spill. In November of 2002, the oil supertanker Prestige sank off the coast of Galicia, Spain, spilling around 60,000 metric tons of crude oil. With the spill, carcinogenic contaminants contained in the crude oil were released into the environment. While the lethal effects of oil spills and oiled animals attract the most public attention and research, sublethal chronic effects have not been researched as thoroughly. With these contaminants dispersed throughout the environment, they would have most certainly spread into the diets of all animals living in the area through direct ingestion or through bioaccumulation in their food web. This led researchers to study what contaminants, specifically polycyclic aromatic hydrocarbons, were present in the blood of avian species, which are greatly impacted by oil spills. The effects of toxic polycyclic aromatic hydrocarbons include inflammatory and toxic effects, immune suppression, and damage to the overall health of individuals. The target species, yellow-legged gull, scientific name Larus michaelis, was used for the study as its plasma biochemistry has been studied extensively along the Galician coast before the Prestige oil spill. This allowed researchers to look at before and after effects of the spill. Additionally, this species has several breeding colonies directly oiled by the oil spill and several colonies in the region that were not. This allowed researchers to sample areas at different toxicity levels to correlate the impact that different concentrations of polycyclic aromatic hydrocarbons have on the health of yellow-legged gulls. With breeding colonies nearby, researchers could also test both adults and chicks to determine if contaminants were ingested directly or if they were consumed indirectly through their food chain. Total polycyclic aromatic hydrocarbons are pollutants found in crude oil that cause damage to various organs when ingested or absorbed. The long-term, sublethal effects of these pollutants on wildlife has not been studied extensively, but researchers have so far found that these compounds cause various inflammatory and toxic effects, suppress the immune system, and diminish the overall health of the organism. The toxic polycyclic aromatic hydrocarbons tested for in the study include naphthalene, acenaphthene, benz at the A position anthracene, tricene, benzo at the B and J position fluoranthene, benzo at the K position fluoranthene, indeno at the 1, 2, 3, C and D positions pyrene, benzo at the A position pyrene, Dibenz at the A and H position, anthracene, and benzo at the GHI positions, pyrroline. Aspartate aminotransferase is an enzyme found in the body, typically at low levels. High levels of aspartate aminotransferase usually indicate muscle or liver damage and typically indicate liver disease. Gamma-glutamyl-transferase is another enzyme typically found in low levels in the body. It is typically used in tests to test for liver damage in the body when at elevated levels. Creatinine is a waste product from the breakdown of creatine in muscles. It is typically used to test for muscle breakdown as well as kidney function, but also can be compared with the results of aspartate aminotransferase as they are in this study to differentiate between muscle damage and hepatic damage. That occurs if aspartate aminotransferase levels are high and creatinine levels are low. Glucose, total protein, and cholesterol were tested to indicate malnutrition or bacterial infections in the population. Like creatinine, inorganic phosphorus levels are compared with aspartate aminotransferase to confirm liver damage in an animal, which would correlate negatively with aspartate aminotransferase. In the study, researchers compared the effects of polycyclic aromatic hydrocarbons in 7 breeding colonies along the coastline of northwestern Spain. The figure here shows areas directly oiled by the Prestige oil spill, represented by black spots on the map. In the figure, the map points out the sample site locations of 3 breeding colonies that were not directly oiled and 4 breeding colonies that were. These samples were taken 17 months after the oil spill occurred. Researchers sampled 95 adults from May 19th to June 5th, 2004, while the birds were roosting on their nest. They additionally captured 135 chicks from June 20th to June 28th, 2004. With each sample, researchers recorded body mass, wing length, and tarsus length. Researchers additionally took a 1 to 2 milliliter blood sample from the ulnar vein. This sample was transferred to plastic tubes and a macrocapillary tube, which were then placed in a cooler maintained at 4 degrees Celsius. Samples were then centrifuged and hematocrit values were determined from the centrifuged microcapillary tube. The researchers separated the plasma from the blood cell fraction and both were frozen in liquid nitrogen at negative 196 degrees Celsius. From these samples, researchers tested the plasma chemicals in 92 adults and 76 chicks. They measured polycyclic aromatic hydrocarbon levels in 62 adults and 54 chicks. And lastly, 39 chicks were tested for both polycyclic aromatic hydrocarbons and plasma chemical levels simultaneously. In order to detect toxic polycyclic aromatic hydrocarbon concentrations in the samples of blood, researchers used high-performance liquid chromatography on blood cell fractions taken from the yellow-legged gull. In each sample, researchers extracted the PAHs using a microwave extraction technique coupled with a 1-to-1 mixture of acetone and hexane to separate the bonds of the PAHs. They then used a deactivated alumina column with hexane as an eluent to separate the PAHs. Finally, they utilized a wavelength programmable fluorescence detector to quantify the levels of PAHs in the blood cell fracture. In order to test plasma biochemical levels in the yellow-legged gulls, researchers used a lambda Perkin-Elmer spectrophotometer to test each sample for levels of glucose, cholesterol, total protein, uric acid, creatinine, inorganic phosphorus, calcium, aspartate aminotransferase, and gamma-glutamyl-transferase. The researchers additionally used data from samples taken prior to the Prestige oil spill in 1997 and 1998 on 2 islands that were eventually oiled by the spill. In these samples, researchers tested levels of cholesterol, total protein, uric acid, and inorganic phosphorus. In the study, researchers found that yellow-legged gulls nesting in colonies with direct oil exposure had much higher concentrations toxic polycyclic aromatic hydrocarbons than unoiled colonies. Additionally, there was no significant difference in polycyclic aromatic hydrocarbons in chicks and adults from the oiled colonies. In reviewing the biochemical parameters tested, individuals from oiled colonies showed a significantly lower levels of creatinine, inorganic phosphorus, and total protein, but had elevated levels of aspartate aminotransferase. The amount of gamma-glutamyl-transferase was not found to be significantly different between males of oiled versus unoiled colonies. However, the levels were significantly different between females. When comparing birds sampled before and after the Prestige oil spill, birds sampled after the oil spill had lower levels of cholesterol and higher concentrations of total proteins. Lastly, birds sampled before the spill showed signs of having weaker body conditions than those sampled after the spill. In adults, the levels of toxic polycyclic aromatic hydrocarbons in the blood showed a positive correlation with levels of aspartate aminotransferase, but did not significantly impact the levels of plasma biochemicals and hematocrit tested. In chicks tested, the levels of toxic polycyclic aromatic hydrocarbons showed positive relationships with gamma-glutamyl-transferase, calcium, and hematocrit. Overall, the study found that TPH levels in individuals differed significantly between oiled and unoiled colonies. They additionally found that certain blood plasma parameters were significantly different in adults from oiled colonies compared to unoiled colonies. These included lower levels of creatinine, inorganic phosphorus, glucose, and total proteins, with significantly higher concentrations of aspartate aminotransferase. I'll cover the significance of each of these in the following slides. Researchers found that adults from oiled colonies had decreased levels of glucose and total protein levels. This is again linked to TPH-induced liver damage, as hepatic tissue is important to both protein synthesis and glycogenesis. While initial reasoning for lower glucose and protein levels were thought to be linked to malnutrition, body condition actually increased after the spill, meaning that the differences were not likely due to diet. In both chicks and adults of oiled colonies, TPAH levels were found to be similar, indicating that they are ingesting crude oil through contaminated food rather than directly ingesting it from drinking seawater. In oiled colonies, elevated AST levels indicated hepatic damage in the individuals in the population. The diagnosis of hepatic damage was confirmed by the low readings of creatinine found in the biochemical parameters tested, as high levels of creatinine would have indicated general muscle breakdown in the body. Additionally, elevated levels of gamma-glutamyl-transferase were linked with TPAH levels. The increase in GGT levels reflect liver disease, damage to biliary ducts, and damage to renal epithelium. This finding could be explained by the female's increased use of their liver during egg laying, as during egg and yolk production, female serum GGT activity increases by nearly 20 times. This result could also be the result of diet difference between males and females, as females would likely increase caloric intake during egg development. Lastly, both chicks and adults had elevated concentrations of TPH levels in their system, but the physiological results reflected more damage to adults. This is likely the result of longer exposure times in adults than chicks. Overall, researchers found that TPAH levels were much higher in birds from oiled colonies than unoiled colonies. These elevated TPAH levels were additionally linked with elevated AST levels for adults and elevated GT levels for females. These findings indicate liver and kidney damage in the oiled populations, which is further supported by the findings of diminished glucose and protein levels, which can occur from hepatic damage resulting in decreased gluconeogenesis and amino acid metabolism. In the study, researchers found that both adults and chicks had elevated levels of TPAH, indicating contamination occurred through the diet and not direct ingestion. Overall, the oil spill has caused damage to the long-term health of the population through chronic contamination. Lastly, long-term studies should be carried out to observe the sublethal effects on the population.